Welcome back to the deep dive. Today we are taking a really close look, plunging right into the tiny details of cancer genomics.
Mm,
We're hunting for those really small but, super impactful genetic fingerprints, the somatic variants, things like SNVs, single nucleotide variants, and indels insertions, deletions.
right. These are the key changes, the mutations that actually drive cancer development.
Mhm.
And finding them accurately, that's absolutely critical for precision oncology.
Yeah, it sounds like the ultimate signal-to-noise problem.
It really is. You got these somatic variants, often at really low frequencies, swimming in a sea of normal DNA you inherited.
Yeah.
If you miss them, or worse, misclassify something normal as cancerous, well, the treatment decisions can go completely wrong.
Okay, so our mission for this deep dive is to explore this pretty interesting new tool called DeepSomatic. It's a deep learning thing, right? Designed to tackle some major hurdles.
Exactly. Two big ones. The historical lack of really good training data and also the limits of the older sequencing methods.
And let's start with that data bottleneck, because you mentioned that's key.
Yeah, absolutely. For, decades, people working on germline variants, the ones you inherit, they've had tons of data, reference sets with millions of known variants to train their tools.
Millions, okay.
But for somatic variants in cancer, the field has just been starved for data.
So you're saying, like, historically, everyone was basically relying on just one main public benchmark set.
That's pretty much it. The HCC 1395, HCC 1395BL set from SEQC2. And it only had tens of thousands of somatic variants.
Tens of thousands versus millions, wow.
Yeah, it's a huge gap. You just can't properly train advanced machine learning models, these AI models, when they haven't seen enough examples of all the different ways cancer can mutate.
Right. If the AI hasn't seen a specific kind of mutation in training, it just won't know what to look for in a real patient sample. Okay, so let's unpack how DeepSomatic tries to fix this. First, why are the standard methods, the short read callers like Strelka2 and Mutek2, why are they hitting a wall?
Well, look, they're really good tools. Don't get me wrong. For most SNVs, they're highly accurate. Strelka2 gets an F1 score, like 0.96 on that standard benchmark for SNVs. That's excellent.
Right.
But they struggle where the genome gets complicated. Repetitive regions, structural complexities.
Because the reads are short, just little bits of DNA.
Exactly. If that little snippet is too short, it might look like it fits perfectly in, say, 5 different spots in a messy, repetitive region.
Ah, ambiguity.
Right. It confuses the caller, leads to false positives, or worse, miscancer variants.
Okay, and this is where, I guess, the long-read technologies come in. PacBio, HiFi, Oxford Nanopore, ONT.
This is where things get really exciting. These technologies give you reads that are just massively longer. We're talking 2 or 3 orders of magnitude longer, tens, even hundreds of thousands of bases.
Huge difference.
And critically, the accuracy, especially for the newer long reads like HiFi and duplex ONT, it's now really good, over 99.9%, so comparable to short reads on a base level.
Okay, so length and accuracy.
Right. The length means they just sail across those complex regions.
Right.
But maybe even more importantly, they let you do accurate, read-based phasing of variants.
Phasing. Okay, we hear that term a lot. Can you quickly break that down? Is it like linking variants together, knowing they're on the same piece of DNA?
That's a perfect way to put it. Yeah. Phasing tells you which mutations happen together on the same physical chromosome strand. With short reads, you might see variant A here and variant B nearby, but you don't know if they're on the same copy mom gave you or the copy dad gave you.
Ah, I see.
A long read can cover both A and B easily, so it tells the caller, hey, these two definitely occurred together on this specific strand. That context is vital for understanding complex mutation patterns.
Context is everything. Okay,
Yeah.
so DeepSomatic, it adapts this existing tool, Deep Variant, which was for germline variants. How did they make it work for somatic? What's the big idea?
The innovation is really clever. It's in how it sees the data. DeepSomatic takes the aligned reads from both the tumor sample and the matched normal sample, and it creates this tensor-like representation. You can think of it like a structured picture, a pile-up image.
Like a side-by-side comparison photo of the DNA at a specific spot.
Exactly. The nodal reads are visually stacked on top, and the tumor reads are right below them in this image. They feed this whole comparative image into a convolutional neural network, a CNN.
Okay, a visual network.
Right. So the CNN isn't just counting variants like older methods. It learns to visually compare the complex patterns, the noise, the alignment signals in both the tumor and the normal samples at the same time.
So it's not just tallying A, T, C, G changes. It's spotting like subtle visual cues weird alignments, patterns across nearby bases, things that statistical models might just miss or call noise.
That's the power of it. It learns the visual signature of what a true somatic mutation looks like, compared to just random sequencing errors or normal inherited variations, because it sees the direct comparison in context, especially with those informative long reads.
Okay, that makes sense. But it brings us right back to that data problem. You build this amazing visual comparison engine, but you need a lot of pictures to train it on. How did they solve that data scarcity issue?
They basically just rolled up their sleeves and generated a massive amount of new data. They got 5 new matched tumor-normal cell line pairs, 3 breast cancer lines, 2 lung cancer lines.
And they didn't just sequence them once with the standard method.
No way. To make it super useful and robust, they sequenced all 5 lines using all 3 major technologies: Illumina short reads, PacBio HiFi long reads, and ONT R10.4 long reads.
Wow. Okay, I just have to pause there. That's 5 cell lines, tumor and normal, across 3 different complex sequencing platforms. That sounds like a monumental undertaking just to get the training data.
It was absolutely a huge effort. But look at the result. They added 263,591 new high-confidence somatic variants to the public domain.
Wait, how much does that compare to before?
It's a 6.5 times increase compared to that single SEQC2 benchmark set we were talking about.
6.5 times more data. Okay, that's a serious jump. And was the diversity important? Did those different cancer types actually look different, genomically speaking?
They absolutely did. And that really validates why they needed multiple cancer types. For instance, the 2 lung cancer cell lines. They had a ton of mutations matching the SBS4 signature.
SBS4, that's the smoking signature, right? The specific pattern of DNA damage linked to tobacco smoke.
That's the one. And that signature was mostly missing from the breast cancer cell lines. So if you only trained your model on breast cancer data, it might really struggle when it sees a lung cancer sample full of those smoking-related mutations.
Right, it wouldn't know what it's looking at. So the diversity makes the tool more broadly applicable. Okay. Performance time. DeepSomatic is trained on this rich, diverse data set. How does it actually perform against the established tools, like CLARS for long reads and Strelka-2 for short reads?
Yeah, the results are pretty striking. It consistently does better across the board, all 5 cell lines, all 3 sequencing technologies. But where it really pulls ahead, where the deep learning visual approach seems to make the biggest difference, is finding insertions and deletions, the indels.
Indels, those are always the tough ones, right? Because they shift the whole reading frame.
Exactly. They mess up the alignment, making them harder to spot reliably. Let's look at some numbers from the HCC 1395 line. For Illumina short read indels, DeepSomatic gets an F1 score of about 0.89 compared to Strelka-2 around 0.79. That's a good improvement.
Okay.
But check out the long reads. On PacBio HiFi indels, DeepSomatic hits 0.815. CLARS, another long read caller, was down at 0.455.
Wow, 0.81 versus 0.45. That's almost double the performance, roughly.
It's a massive leap in reliability for finding indels with long reads. Really significant.
Definitely. Now, you mentioned this multi-cancer boost. What's the tangible benefit? How much better is the model trained on all 5 cancer types compared to one trained just on that original single cell line?
Right, they quantified that. And it's impressive. The model's trained on the diverse multi-cancer set compared to just the single HCC 1395 line. They cut down the total number of SNV errors by almost 40%.
40% fewer errors. That's substantial.
It is. But hold on. The indel improvement is even more dramatic. The multi-cancer models reduced indel errors by, get this, 95.2%.
95% fewer in indel errors. That seems almost too good to be true. What's going on there? How does seeing different cancers make it that much better at finding these tricky variants?
It's a great question. It suggests that the model isn't just learning cancer mutations. It's learning to recognize and ignore the specific noise patterns and technical artifacts that come from different sequencing technologies, different sample preps, maybe even different underlying genomic backgrounds in the cell lines.
So by seeing more diverse problems in noise, it gets better at identifying the real signal across different contexts.
Exactly. It learns robustness. It becomes less easily fooled by, say, a mapping glitch that looks like an indel but is really just a technical issue common in one type of data.
Yeah.
That huge drop in indel errors suggests it learned to be much more discriminating.
Okay, that makes sense. Robustness is key, especially for real-world clinical samples, which are never perfect.
Yeah.
Did they test DeepSomatic on messier, more realistic scenarios?
They did. They simulated varying levels of tumor purity, how much actual tumor DNA is in the sample, going down from 90% to 60%. DeepSomatic held up better, maintained higher F1 scores than the other callers across those levels.
Good.
And they also tested against contamination, something called TIN, or tumor-normal.
Tumor-normal, meaning some tumor cells accidentally got into the normal reference sample.
Precisely, which makes the comparison much harder, obviously. And yeah, DeepSomatic showed more robustness there, too. These things happen in real labs.
For sure. That adaptability is vital. Okay, but what about situations where you just don't have that matched normal sample? Sometimes it's just not available, right? Cost, or maybe just not enough tissue.
Yeah, a very common scenario. So DeepSomatic has a tumor-only mode for that. Obviously, without the matched normal, it can't be absolutely certain if a variant is somatic or just inherited germline.
So how does it try to figure it out?
It uses other sources of information as filters, primarily a panel of normals, basically a big database of common variation seen in healthy people, and also population allele frequency data.
Okay. So if a variant shows up frequently in the general population or in that panel of normals, it assumes it's probably inherited, not a new cancer mutation.
That's the logic, yes. The tumor-only models using this approach get pretty good recall, meaning they find most of the true somatic variants. The precision, telling true from false positives, is a bit lower than the paired mode right now, but it's still very useful.
That's still a big deal for those cases. What about really challenging samples? Like the old archive samples stored in formalin, FFPE.
Ah, yes, formalin-fixed paraffin-embedded tissues. Standard practice for storing clinical samples for years, but the formalin really damages the DNA. It creates specific chemical damage patterns that can look exactly like mutations to a variant caller.
Tricky.
Very. But DeepSomatic was adapted for this too. They tested it on FFPE whole genome sequencing data, and it got an F1 score of 0.88 for SNVs. Significantly better than Strelka-2 and Claris on the same FFPE data.
That's huge for unlocking all that historical data sitting in pathology labs.
Absolutely opens up vast archives for research.
And one last check, cost is always a factor. Whole exome sequencing, WES, just sequencing the protein coding bits is still common because it's cheaper. Does it work there too?
Yep, they covered that base as well. They have WES and even FFPE WES models, and those also outperform the other callers on that type of data. So the core CNN approach seems flexible across different sequencing strategies.
Okay, so let's zoom out. What DeepSomatic seems to have achieved here is, wow, pretty remarkable. A highly accurate deep learning tool for finding these crucial small somatic variants. It works with both short and long reads, and it adapts to tough, real-world situations like having only the tumor sample or dealing with degraded FFPE DNA.
That sums it up nicely. The tool itself is a major advance, but I think the maybe even bigger contribution long-term is the data they generated and released.
Right, that 6.5x increase in benchmark data.
Exactly. By creating and sharing these high-quality, validated benchmark sets across 5 different cancer types and 3 technologies, they've fundamentally raised the bar for everyone developing these kinds of tools in the future. They've helped fill that critical data gap.
That public resource is huge, yeah. It should really accelerate the whole field. Okay, so here's the final thought I want to leave you, our listeners, with, drawing from the research itself. Even after generating this massive new data set, this huge leap forward, the researchers themselves noted they are likely, and I'm quoting loosely here, quite far from reaching diminishing returns on training data.
That's such an important point. It's humbling,
Yeah.
really. It tells us that cancer is just so incredibly diverse. The mutational signatures vary wildly, not just between cancer types, but between individuals, even within a single tumor.
So even this massive data dump is just scratching the surface.
It suggests that, yeah, if we want truly universal, truly comprehensive diagnostic tools that capture the full spectrum of human cancer genomics, we're going to need vastly more data than even this project generated. We've only just begun mapping this incredibly complex landscape.
The search for that perfect cancer map definitely continues, and it looks like it's going to require an even bigger data effort than we might have thought. Thanks for joining us for the deep dive.